All right, set number two notes in our unit on population genetics. Yesterday we talked about the fact that you have ways to calculate a loop frequency utilizing genotypes. A lot is based upon the concept of what's called the Hardy-Weinberg equilibrium law. This is basically a theoretical, perfect, ideal population where everything works mathematically like it's supposed to. In real life, it's probably not going to happen. There's a lot of assumptions that are made in order to make this work, and most populations violate those in some way, shape, or form. But why would we want to know about an ideal population? Well, if you think back to statistical analysis, a lot of the models and different kinds of tests you can do use an expected versus an observed. Well, this would be our expected values. This is what we expect to get, so we'd be able to calculate it utilizing the expected values. So it's used to determine genotype frequencies that we would expect in a perfect ideal population. It's named after Gottfried Hardy and Wilhelm Weinberg, who both came up with the idea independent of each other. One was in England, one was in Germany, and they came up with this idea mathematically of how population would work. Since they both published around the same time in different parts of the world, people give them credit of being co-inventors of this. It makes a number of assumptions. This is why it's an ideal, perfect population as opposed to a real population. Number one, the population is infinitely large. Most populations aren't infinitely large. They have a finite level. Mating occurs at random. That means there is no mating selection. There is no people choosing partners that they want. It's just random matings. That usually doesn't happen in real life with any sort of organism. It's usually going to be very selective towards the types of things that they do. And when you choose a mate and things like that, that's going to throw that off. There's no selective advantage for any genotype. What they're saying there is, it doesn't matter whether you have big A, little a, big A, little a, or any other variation in a population, because all of those things have equal chances. They're all equally perfect the way they're supposed to work. It's not like one of them is better for your environment, so therefore it works more often that one becomes an advantage. There's absence of other factors such as there's no mutations, because if you mutate the population, it throws off all the frequencies. So you can't have mutations in a population. We know in real life, mutations occur all the time. There's no migration. All organisms in a population stay put. That doesn't happen as well. Everything migrates in some way, shape, or form. There's always movement around within every population. There's no genetic drift or random chance the population goes one way or another. The idea of pulling it out sort of randomly out of a hat and just sort of moving everything to one direction as opposed to the other. If I pull out a 50-50 bag of red and blue marbles, chances are I should get roughly 50-50, but I could get a handful of almost all blue marbles and push the population in one direction. That would be genetic drift. The formula for it is based off of that binomial theorem probability, P plus Q equals 1, except we're taking a different step forward. If you remember back to Punnett squares in middle school, you had your big A, little a, big A, little a, big A, little a, little a, little a, if you took two heterozygotes and crossed each other. So that's the ratio you got. 3 dominant, 1 recessive, or 1 to 2 to 1. The reality is, the math behind that is very simple. If this is P, this is Q, you can just fill in the square as a matrix. So big A is going to be P squared. Big A and little a are going to be P and Q, and there's 2 of them, so it's 2PQ. And then little a is going to be Q squared. That's how you determine the math behind the Punnett squares. So the formula is basically P plus Q, binomial theorem, squared, still equal to 1, because still the overall value is still 100%. And I'm going to get P squared plus 2PQ plus Q squared equals 1. So if I know P plus Q equals 1, and I know those values can figure those out, I can solve this way or vice versa, go back and forth, as long as I have that information. So these are all ideal expected numbers that I get from this problem or process of doing this. So let's start with perfect. P is 50%, Q is 50%. So therefore my values would be P squared, 0.5 times 0.5 is 0.25. Same thing with Q squared. And then these two are both 0.25. There's 2 of them, so it's 0.5. So there's my values in an ideal, perfect little Punnett square. So let's do a practice set. Make sure we're on the same track and understand what we're talking about here. So I have a population of rabbits. Brown ones are dominant. White ones are recessive. Brown rabbits have the genotypes of big B, or big B, little b. White rabbits have genotype of little b. And the frequency of the big B genotype is 0.35. I already know something about that though, because do I know the value of P, the frequency of the brown allele? No, because I don't know the other values. But I do know p squared because that is big B, b times b, p squared. So p squared is going to be equal to 0.35. So if I want to figure out the value of b, and I know that's 0.35 is p squared, that's going to be p squared is equal to 0.35. That means for me to find p, I just take the square root of 0.35. And that'll give me my answer. That answer is going to be 0.59. 0.59, if I know p plus q equals 1, 0.59 is p. That means q is 1 minus 0.59, which is 0.41. So if I want to figure out what the value of b, big B, little b is, that's heterozygous. If I go back to my equation, that's the middle equation, it's 2pq. I know p, I know q, I take them times each other, I take that times 2, and for the answer I get 0.48. So I know 0.35 is this one, I know 0.48 is this one. I can just calculate my third one for little a by just adding them together and subtracting from 1. Or I can take that squared and get the same answer. And that's how we get the third part of it. So practice set number 2. Hypothetical population of humans, there's 10,000 humans. 6,840 individuals have big A. 2,860 individuals have AB. So I don't have to have the straight big A, little a. I can also do this in the same form this way. I have to change my value a little bit. There's still an A and a B. Those are my two alleles. 3 individuals have the blood type big B. So this is going to be my p squared, my 2pq, and my q squared. So I actually know my genotype values. But I have to turn them into percentages. The easy way to do that is 2,860 divided by 6,840 will give me the percentage for that. Sorry, my bad. 6,840 divided by 10,000, so that's just going to be 0.68%. And this is going to be 0.28 or almost 29, and that's going to be 0.03% because it's just out of 10,000. So the frequency of each genotype, well, that was the easy way to figure it out. 0.684, 0.286, 0.03. So that would be my values right here. Just taking the 10,000 divided by those. There's a couple ways I can figure out A. I could do the previous equation we did the time before. I could take 2 times this plus, or 2 times this number here plus one of those divided by 20,000 and get this. Or I just realize that's p squared. So I take the square root of 0.684 and I get 0.83. If I know 0.83 is A, then I just take 1 minus 0.83 and I get B, which is 0.17. That's all I have to do. You basically go back and forth between the two equations, utilizing them to solve these problems. So the next question here is going to be, there's 25,000 individuals in the next generation. How many would be big B? Well, if I want to figure out my percentage of big B, it's up here, right? 0.03. Well, if I take 0.03 times 25,000, that'll tell me how many individuals I should have that if the ratio stayed the same, because there is no those other factors that affect it. It's this Hardy-Weinberg equilibrium. In that case, there would be 750 individuals in the next generation because there's more people that have that particular trait. And that's how you calculate that particular kind of a ratio.